Hello to everyone who's joined, and welcome to the 22nd episode of CCLG's Research Talks for Parents and Public. I'm Ellie Ellicott, and my job is sharing research with our supporters. So let's start with some housekeeping. We'll be holding a Q&A session at the end of the talk, so please do submit your questions whenever you think of them using the Q&A button at the bottom of your screen. If you'd like to use the chat function, please make sure you set the to drop down to everyone, or people may not see your message. The webinar is being recorded and will be available publicly on our YouTube channel, so we'll send out the link when it's ready to watch. This month's talk is called Tackling Childhood Lymphoma from Technology to Trials and is being led by Professor Vikki Rand. Vikki is the director of the National Horizons Centre at Teesside University, which connects researchers with industry, the NHS and more to make a real-world impact. She also conducts research into lymphoma with the goal of finding ways to make treatment safer and more effective. So, without further ado, I'm going to hand over to Vikki and awkwardly share my slides whilst you start talking.
Yes, thank you very much, Ellie. Yes, so slight technical difficulties, so we're doing a good impression of Chris Whitty. So yeah, thank you everybody very much for coming. I'm really looking forward to presenting some of the work that my lab and the collaborators have been doing around paediatric lymphoma. Are we good to go?
Yep, sorry, it's still working on it.
Okay. Yeah, so I'm going to be looking at technology, and I'll just tell you a bit about myself. My background is on cancer genomics. I did my PhD on the Human Genome Project many years ago, which is the equivalent of walking on the moon in biology terms. And since then, I've been using technology to really kind of push what we can do to understand cancer across different types of cancer and also different types of samples and also different clinical settings. So the work I'm going to present has been supported very much by CCLG. I mean, without CCLG, we would not be able to do this particular study. Also funding from Little Princess Trust, a Bennett Fellow, personal fellowship from Blood Cancer UK, and then also from NEPAC, which is a Northeast cancer charity. Next slide, please. If you hit it twice, just to save your finger. Actually, do it two more times. And once more. I may have to do the whole thing. Yep. So, oh, sorry, go back. Okay, perfect. We'll be perfect at the end of this. So basically, you may already be aware, but I thought I'd set the scene with some of the background around cancer in children and also in terms of lymphoma. So, I mean, they start with a positive. More children, young people than ever before are surviving cancer. And a lot of this is down to kind of the work of CCLG and a number of our collaborators across different cancer types. More than 8 in 10 children, young people are now surviving for 5 years or more. Some cancers and types, subtypes within them have much lower survival. But it's estimated that there are 45,000 people in the UK alive having been diagnosed with childhood cancer, and approximately over 1,000 are added to this number each year. But as you look, there's a number of different types of childhood cancer. There's more than 200 different types across brain and spinal, leukemias, liver, neuroblastoma. And each type has its own name and test to diagnose exactly what type of cancer it is to ensure that doctors plan the right point of treatment. But also within each subtype, we also have more and more different types. And what we want to do is understand the biology to identify the right treatment for the patients. If you can, next slide, please. I think if you hit it twice. Yeah, 2 more times. Yeah, and 1 more. Perfect. So basically, what is lymphoma? So this is information that's come from CCLG and CR UK, and they're really, really good resources to kind of understand the different types of diseases, but also the types of treatments and other key information. So lymphoma, for those who are aware, it's a type of blood cancer. It develops when the white blood cells who are there to kind of protect our body abnormally grow. It's called cancer of the lymphatic system. The figure which shows the green is the lymphatic system. It starts in the lymph nodes. You can see some of those highlighted on the figure, but also in other organs of the lymphatic system, such as the bone marrow and spleen. And you can see where the location of the spleen, the thymus, and tonsils are key areas. They're basically, it's the third most common type of childhood cancer, and there's about 1 in 10 new cases. Well, I count about 1 in 10 new cases of cancer each year. There's probably about 80 cases of lymphoma and B-cell non-Hodgkin lymphoma per year. Next slide, please. Perfect. So in terms of lymphoma, I was talking about how complicated it gets. We've got 200 at top level by organ, but also kind of different subtypes. And this is the WHO classification of BNHL, and each cancer type has this classification. This is what pathologists and clinicians use to diagnose the different types to develop the treatment pathway for each patient. So you have broken down into kind of cell type, and I think one of my collaborators, Suzanne Turner, has previously done a talk and talked about B and T cells and some of her research linking into particularly the T cell area. But we also have non-Hodgkin and Hodgkin lymphoma. And if you hit the slide one more time, next, whoop, yeah, perfect. These are the two types that I mainly work on. They're both B cell non-Hodgkin lymphoma, diffuse large B cell, and then Burkitt's lymphoma. Next slide, please. And looking across the different subtypes of B-cell non-Hodgkin lymphoma in children and adults, you can see from the 0 to 14 years that Burkitt's lymphoma, apologies for the resolution, this is taken from a paper, there's a few where the PDFs are not the best quality, but Burkitt's lymphoma account for 38% of cancers, lymphoma cancers in the kids. If you look at your teenagers, so hit the slide one more time, you can see that diffuse large B cell is becoming more common with 37%. And if you look in adults, the next hit one more time, perfect. You can see that actually follicular cancer is becoming the most prominent subtype within that ages. So you can see the differences in the types of subtypes that emerge across the age spectrum. If you hit one more time, yeah, perfect. So you can see that Burkitt's and diffuse large B cell in children up to 0 to 14 of 38%, 20%. And in teenagers, you can see this is slightly changing from 21% with Burkitt's, 37% with DLBCL. But you can see a real change in adult population where Burkitt's is a really, really small fraction of the different subtypes. So we're already seeing differences across the different age groups in different types of cancers. Next slide, please. I think we hit this 2 more times. Yeah, perfect. So Burkitt lymphoma is the most common pediatric BNHL. This is a really aggressive, fast-growing cancer of the B cell types of lymphocytes, the white blood cells. It was first described by Dennis Burkitt in 1958. He was an Irish surgeon and a researcher who, while working in Uganda, described a new form of childhood lymphoma. And this was described across equatorial Africa, and he is looking at a map of Africa, and he defined the Burkitt belt, which goes across the continent, where there's a kind of a link with malaria and EBV. While he was there, he'd identified a number of cases of children coming with large jaw masses, predominantly in the jaw and also in the abdomen. But his work there defined this new subset, which was named after him, but also showed that chemotherapy alone could successfully treat the cancer. Next slide, please. We also know that the sporadic Burkitt lymphoma, which occurs in the UK and Western countries, looks very similar to the African Burkitt lymphoma that Dennis Burkitt described. And what does this look like? So down a microscope, this is what a Burkitt lymphoma looks like. So this is what a pathologist is looking at once they've stained the cells. And if you hit the slide once, please. Yeah, so you can see this dense background of small monotonous malignant B cells. This is the sky. And then the next slide, yeah, thank you. And then also you can see the scattered lighter stained macrophages with engulfed, what I call apoptotic debris, which is basically they're munching up, eating up the dead cells in the tumor. But what you see is a real blanket of really, really similar looking cells. Next slide, please. So what about outcome of TAMP? We know what it looks like. We can define it. How do we actually treat it, and how are the studies going? I'm going to be showing you a number of these particular graphs all the way through it, and they're called Kaplan-Meier's. You may have seen them before in other studies, but this is how we look at survival. So once we've diagnosed the patients, we can understand how patients are responding to treatments. Hit the slide once, please. You can see on this, so on the left-hand side, that's the probability of percentage of survival of children in these particular studies. You can see by this drop in the arc that most events occur early. So these are patients who are either relapsing or not responding to treatment. But you can also see that if you hit the slide one more time, that there actually is an increase in survival with each trial. So the very bottom, you can see the horizontal slide from 1977, this is CCG 551. This showed that most children had about a 45% chance of survival during this study. But as you kind of go up to the very top ones, where you have the CCG 5961 and the COG study, you can see this is almost at the well over 90%. So over this period of time, you can see there's been such a change in how treatment and response to treatment. Next slide, please. However, there are still some clinical challenges. So because we've got 90% children who are diagnosed with BNHL, They respond really well to current treatment. However, the treatment is really intensive and has a lot of nasty side effects. And also, if children don't respond to treatment and have what we call refractory or relapse disease, the survival is really poor. So what we wanted to set out is to understand how could we investigate these areas to kind of really improve the treatment and for each patient. Next slide, please. So our research question, research question in my lab is, what do we set out to do? Basically, we wanted to look at two cohorts, so one from the UK and one from Africa. So looking at, when we look at burkitt's, the sporadic disease, which is seen in the UK, and the endemic burkitt's, which is seen in kind of Malawi and Africa. In the UK, we have over 90% survival. In Malawi, we have about 50 to 70% survival. The treatment regimes are very different based on the clinical setting, but we can look at the basic biology and looking at that blanket of cells and unraveling that to see what the differences and similarities are. So if you hit the slide once, we wanted to identify who were under-treating, and then next, who were over-treating in the UK. Next, please. And who, in terms of the relapse patients, how can we treat those successfully? And then one more time. And also see whether within the Malawi African cohort, are there some children who can also be similarly treated as those in the UK who relapse? Next slide, please. So we start out by looking at the UK cohorts, and this is why the CCLG has been so important and so valuable to this research. So across the different CCLG centres, now banked within the Vivo Biobank, patient samples and clinical data are all collected. So we've got patient samples from across, all the way from Aberdeen all the way down to Southampton and everywhere in between. Next slide, please. When we got the patient cohort, we had the pathology reports. These were reviewed for each sample just to check it had the right diagnosis. Next slide, next, please. Yep, we collected patient samples and also used this for pathology review. So you can see here, this is what they essentially look like. We get a little block which has a little bit of the tumour in there. We take a slice of that tumour and stain that on the slide. And this is what will look like down the microscope with the starry sky. And also we get frozen sections as well that are put into -80 for storage, and we can use them to get really good quality DNA. Next, please. And then we use this material to do diagnosis, so based on the WHO classification and any other markers that have appeared in the literature since its publication to really ensure we're diagnosing and studying the right disease. Next, please. And then it's important to collect that clinical data, and this is required for that survival analysis. So we looked, and this is actually a table from our paper, where we had looked at kind of the sex, age, a diagnosis, time to relapse, bone marrow and other involvements that are all part of the clinical decision making. Next, please. And I can't say going really further about saying thank you to everybody who did this work, because without the nurses, the whole CCLG Vivo team, clinicians, lab staff, and everybody who is committed, we could not have pulled this information out. It's a very complicated process to get this information across all those different centres. So it's a pause to say thank you. Next slide, please. In terms of the Malawi cohorts, actually hit it twice. I think. Once more. Perfect. So we work with a hospital in Malawi called the Queen Elizabeth Central Hospital. The clinical team at Newcastle NHS Trust at the kids wards, they have set up a charity, and they've been working with this hospital for decades to really help develop the protocols, which is why the protocol in Malawi has really changed from very few patients surviving through to kind of up to 70%, and this is obviously still growing. Malawi is one of the poorest countries in the world, but we have this dedicated paediatric ward which has been open since 1997. And importantly, this medical treatment is free. They do have a private healthcare system, but this is a free hospital, and it supports a lot with children with cancer in Malawi. Next slide, please. Yeah, once more. Yeah, perfect. So this is what the hospital looks like. So this is the paediatric ward, and what types of patient samples we get from there are taken as kind of through a syringe, like a bone marrow, and this is stored in a solution, and this fixes the cells for us, similar to the FFP bot, but kind of means that can sit them on the shelf for however long they need until we can get them back to the UK. In terms of the diagnosis, this is Professor Liz Molyneux, who is an absolutely amazing clinician, and she does fantastic work with the hospital in Malawi in collaboration with the Newcastle team. So this is part of the one on the labs. We also have a real-time pathology. So samples are taken and stained there. And then the team in Newcastle are actually looking at these remotely in the UK and giving a diagnosis. Next slide, please. And I think 2 more times. Yeah. So in terms of these what called fine needle aspirates, the sample at the bottom, see the very brown bit at the bottom, those are the kind of the cancer cells that we're looking at. We look at immunocytochemistry, so this is that WHO classification, and we also look at other markers, and I'll go on to kind of what FISH is in a moment. And then we also extract that DNA, when this is a gel showing the different bands show the different sizes of DNA that we are extracting from the different samples, and that's how we'll move on to how we analyze the data. Next slide, please. So for us, it's what did we do? It's basically studying the DNA. So we wanted to look at all these lymphoma samples and really unravel what was happening at the very base level. And I'll go into a few slides about DNA and some of the techniques that we used. Next slide, please. Maybe twice more. Perfect. So basically, for those, I'm slightly obsessed with DNA. I think it's both from doing my PhD on Human Genome Project. DNA is the book of life, and this is the molecules that really carries out genetic instructions. You may be familiar with Watson and Crick, who discovered the structure of DNA, and you may also be familiar with Rosalind Franklin. These are two papers that were published back to back in 1953. And Rosalind Franklin's work, you can see the kind of the figure with the cross on. This is what we call X-ray crystallography, and this is looking down on the DNA, and this helped Watson and Crick to understand that the DNA was a double helix in that structure. Next slide, please. So basically, when you have the DNA, you also have chromosomes. So chromosomes are from chromosome, well, basically named in size, so chromosome 1 through to 22, with 1 being the biggest and in the 20s getting smaller. But we also see within the cancers that things go wrong, and chromosomes kind of swap bits of information. And one of the hallmarks of Burkitt's lymphoma that really helps us to identify that we're looking at the right patient samples is what we call the MYC translocation. A majority of these cases happen between a bit of chromosome 8 and chromosome 40, that's switch material. I've hit it twice, some arrows should appear, yeah. And what you end up with in your genome is basically the four chromosomes, but we only have one normal chromosome 8 and one normal chromosome 14, but you end up with a slightly smaller chromosome 8 and a slightly bigger chromosome 14. And this causes a fusion of two bits of the genome, which makes the MYC gene behave badly and deregulated. And this is a real hallmark. And how we look at this is on the next slide. So we use FISH, and then once more. Yeah, so I'm also training everybody to become DNA and FISH experts at the end of this as well. So, but this is kind of one of the really cool ways of looking at it. So we can pull the DNA apart and we can use fluorescent tags to kind of highlight different bits of the genome. And this is where we've gone those bits of chromosome 8 and chromosome 14 and coloured them. So MYC, chromosome 8 is in red, and the green is IGH, which is on chromosome 14. On the top one, you can see that these two probes, these two colours are separate. This shows that this is normal. There is no MYC translocation in this sample. But on case 2, you can see that there's a green and a red. These are the normal chromosome 8 and chromosome 14, but you can also see the kind of green, yellow and red patches, and this is showing that translocation. So for us, it's confirmed that it's the right type of case for us to be studying and moving forward. Next slide, please. Hit once, twice, maybe again, yeah. We also know that there are different types in the chromosome, that different bits of the genome has potential impact on prognosis. And this is showing again, the Kaplan-Meier's. So you can see that the probability of survival on the left-hand side, and then the kind of plateau. And you can see from the red and the dark blue lines on both figures that the abnormality is actually showing a poorer survival for the children in this study. Next slide, please. Yeah, and we can look at this in our cohort as well. So it's finding all of those, kind of pulling all the pieces of the puzzle together, and again using FISH. So this is showing that we have a gain of chromosome 7. So we've got 3 green dots and 3 red dots, and this shows that we've got extra copy of that chromosome. We can also show that we have deletions of a chromosome 13 on the right hand side, showing that we've got one red and 1 green dot. Next slide, please. And we also know from previous studies that if we look at karyotype, which is looking at things at a very big level, so looking across each chromosome, and if you've been to any of the talks from the leukemia group at Newcastle University, they've used cytogenetics for defining leukemia, analysing that and made significant advances in that area. Very similar technique. So basically here we're looking at different types of karyotypes, and we know that from this that basically if you have a very complicated chromosome structure, so you have lots of extra bit copies or bits that have moved around, and the MYC complex, which is that pale green line, that patients do worse. So we've got a lot of signs showing that our DNA and looking at DNA in these cancer samples is really complicated, and that is something which is causing patients to have a poorer outcome. Next slide, please. So problem with these studies is their studies are small and basically will these findings still be seen in other and larger cohorts? And also what about the rest of the DNA? FISH and cytogenetics are only really giving us a snapshot into the cancer. So next slide, please. And this is where I get very excited because the Human Genome Project, I mentioned at the very beginning, this enabled us to sequence every single base. So all 3 billion bases, 3.6 billion bases of the DNA. Just imagine it as a long piece of string of beads. But this gave us the technology to look at that and every single base. And it's now referred to as that reference sequence. So we basically can use that and we can look at any tumor sample and compare it to that reference. And essentially, we're paying spot the difference. So we're looking for kind of all those things that are different in the tumor compared to the normal DNA. And if you're interested in going to see in the book, the Wellcome Trust Museum down in London, they actually have every chromosome, all the bases printed out to be able to see there, which is quite exciting. Next slide, please. One more. So what we can do now with the technology that's come from the Human Genome Project. So this is called an Affymetrix gene chip. This is basically has all of the genome on this chip, and we can basically look across all the different chromosomes as we did with the fish, but basically every single base across there to look for abnormalities and changes in the chromosomes. And if we look at these two samples, so we have the top one and then the diagnostic sample, and we have a matched relapse sample. And for all of those eagle-eyed, you'll be able to see as you kind of go along and taking the relapse sample that you've got some areas that are slightly high above the bar. If you could hit the slide one more time. Particularly on chromosome 7 and chromosome 12. This shift from 0 shows that we've got a gain of 7 and a gain of 12. And what we can do with this data is actually zoom in to even smaller areas to identify what genes are in there. So you can see how the technology is developing. Next slide, please. We also use Sanger sequencing. So this is what the human genome was based on. We take a fragment of the genome and these machines and basically can look for particular single, right, I've done every single base, a change there. So here you can see that we have a C and a T in that kind of area there, and that's showing a mutation. We can see the resolution we're getting down to. Now sequences are tiny, they're about this big, nanopore is tiny, and basically you can kind of literally do sequencing at the bedside now. Next slide, please. And this just shows the evolution of the types of equipment and the types of tools that are available. So starting on the top with WHO diagnosis, and as you go around, we can look at all sorts of things that are happening in the DNA and also other layers within that kind of cancer cell. And companies like Illumina, Agilent, NanoString, and Nanopore are ones which are really pushing the boundaries in terms of the technology that we can use. And this is really what my lab focuses on. We basically use that multi-omics approach and that data modeling to understand cancer genomes. Next slide, please. So what have we found? So with the project with the CCLG cohorts, we have found the first molecular biomarker in pediatric B-cell non-Hodgkin lymphoma. This is a paper that we published a few years ago, which was a collaboration. Again, you'll see that Suzanne Turner is on there as one of our key collaborators. You can see how we work really closely together across that CCLG community. Next slide, please. And basically, we identified mutations in this gene in almost 50% of samples, which is a very high percentage of cases that actually have this mutation. This figure shows the different colours represent different parts of the gene, and within here, those different shaded areas do different bits within that structure. So if you have a mutation, it might be that the endpoint, which is called a protein, has a slightly different structure and doesn't behave in the same way, or it could actually be manipulated to be able to be targeted by different therapies. So by having these mutations, it shows that there's something here that we need to investigate further. Next slide, please. And just to show what it looks like when we're looking at the data, so this is whole exome sequencing. So this is sequenced across every single gene across the genome. This is using a tool called Integrated Genome Viewer, where we're showing cases with a p53 mutation. And you can see that we have a long line of red, which is a T, and this is showing that the normal base has been replaced by a T in this sample. And that's what is then summarized into that figure. Next slide, please. And also deletion. So this is from the Affymetrix array. So that data that you saw earlier showing the different copies and losses. And this is summarized into this figure. And it also shows that we have deletions of 17p in 15% of cases. Next slide, please. So when we put this all together, we wanted to look at kind of the risk groups based on the p53 status. So if we look at the normals, we looked across to see they didn't have any, this you see at the very bottom panel, they don't actually have any 17p or p53 abnormalities. But as you kind of move along, you've got kind of monoallelic, where you can see there's a number of mutations. But the interesting area is that biallelic. This is where we mean that both chromosomes have either an abnormality in some way, which actually is completely stopping that gene in its tracks from doing its normal function. So next slide, please. But what was interesting about this particular gene is when we put all this data together and looked at the survival, we identified that any abnormality with P50, a sample with P53, had a worse outcome. And interestingly, that none of the 39 cases that were reported to have disease progression or relapse had a P53 abnormality at all. So this made us want to look at this data further to kind of really understand and get into the core of what's happening with this particular gene. Next slide, please. And this is where that biallelic, monoallelic, and normal come into play. So we can see that the green line normal, so that's no abnormality on 17p or p53, 100% survival. But when you start looking at monoallelic, so one of the chromosomes has an abnormality, and then the biallelic, the red, shows that a much worse outcome. Next slide, please. But also we wanted to look in the current situation where rituximab now is part of the treatment protocol. A lot of our samples were before this particular drug was introduced. So we wanted to see whether this phenomenon that we identified was also identified in these cohorts. So we looked at this particular study. So by using the definition of what high-risk patients are in this particular trial, we wanted to see whether we could differentiate our data to see whether p53 still would have an impact in this particular cohort. Next slide, please. So this is basically just seeing whether our data looked right. I mean, sometimes when you look at data, we need to make sure that it's behaving as we expect it to behave. So this is showing kind of high risk group is behaving, it basically has a worse outcome than the low risk group. Next slide, please. But what we could see is the answer is yes. So we can show that the patients with p53 abnormalities did have a worse outcome within this particular group as well. So even though rituximab era, p53 is a really powerful marker for us to kind of be exploring further in terms of kind of leading into clinical trials. Next slide, please. But we also wanted to look at what happens in the relapse samples. So most patients, if have Burkitt's, either relapse, have refractory within that kind of very, very quickly, within 6 months to 1 year. So biopsies aren't often taken, but within our cohort, we did have access to some relapse cases. But we also looked at what was in the literature. Can you put it one more time? Yeah. And what you can see here is a cohort of 23 patients where you have the initial diagnosis, and the yellow shows that there's an abnormality in that region on that chromosome. So you go from chromosome 1 across to 21. And then you look at the sequential karyotype. So that's from the same patient once they've relapsed sample. You can see that 17p is coming up as being altered, but you can also see the complexity within that genome. Next slide, please. So that gave us some interested in terms of whether 17p was going to continue to be an important factor in relapse, because if it's not maintained, then we don't understand that the biology is more complicated. But if it is maintained, we know it's a really important gene for us to be looking at therapeutically of how we can actually target that. So if you can hit once more, please. So this figure, if you look at this line, hit it again, please. Yeah, so if you look at this particular case, you can see that the diagnostic was marked D and the relapse called P. They both have abnormalities and basically biallelic abnormalities at diagnosis and relapse, showing the importance of P53. Hit it a couple more times. Yeah, but in the BL23, this shows that's yellow, so we've actually got monoallelic diagnosis, but as we progress into relapse, it actually becomes biallelic. So this particular clone and type of cell is dominating, and again showing that importance and the play that P53 is having. Interestingly, when we looked at diffuse large B-cell lymphoma, we saw monoallelic only within the diagnostic and relapse pair. So looking at a slightly different mechanism in terms of that kind of sustaining a relapse to be looked into. Next slide, please. But also, what about the endemic Burkitt lymphoma? So if you think back to kind of the little jelly bean men, jelly babies, we've got the Malawi case where we wanted to identify, do we still have that really high risk subgroup? We've defined that using P53, but unfortunately, we didn't see this as significant in that endemic cohort. But this could also be because of the different kind of treatment protocols. So there's still more there to unravel. And basically, you can see that from the blue and the red lines are just right next to each other, so showing no real difference in terms of abnormality. So there's further work to be done on that cohort to identify the prognostic markers. Next slide, please. So what did we do? We basically went back to look at the literature to identify what do we know about pathways deregulating. So during this project, there was a number of big studies that came out looking at different mutations, not associated with survival, but looking at different pathways that we could be using for different therapeutics. And this is kind of showing the BCR receptor, which is the kind of the Y shape at the top, that can be targeted. Leads into what we call the PI3 kinase pathway, and there's a number of members of that pathway and those genes that could be targeted. So what we wanted to do was look at our data to see what the mutation rate was like and whether they were abnormal in our pediatric cohort. Next, I think one more time. Yep, so one of the ones that came up was FOXO1. Next slide, please. So when we looked at our data, we identified that there was actually a high frequency of these FOXO1 mutations in both our sporadic and our endemic cases, and higher in our endemic cases at 54%. Next slide, please. And when we looked at this data in more detail, we identified, you hit it one more time, please? Yeah. That we actually found this cluster around what we called the AKT recognition motif. Not easy to say, but we've got a real structure around this part, which has a functional impact on how the gene operates in its normal capacity. And the fact that we had both the endemic and the sporadic clustering in this area, we wanted to have a look at what would be the impact if we kind of use technology to remove this part and look at the function of the abnormal gene. Next slide, please. We also looked at clinical relevance, and basically there was no clinical in terms of the survival in terms of kind of FOXO1. Next slide. Sorry, next slide. Yeah, perfect. But what we did do was using CRISPR-Cas9 gene editing. So this is taking looking at the DNA and taking a short fragment of the DNA and using this to silence the gene. So we looked at a number of different pieces of that DNA to silence the gene. Of course, we can see on the blot there. So if you have FOXO1, you have the big thick bands. This is looking at the protein level. But when you have SG1, SG2, SG3, they are the ones where we're actually going into the gene and silencing it to look at the function. And as you can see, that band is really, really faint. So we have silenced most of that gene in there. And when we look at the data in terms of looking at the expression of the control versus the FOXO1, so the graph at the bottom with the blue and the red, you can see there's a significant difference in terms of the kind of function of that gene. and it's a potential in terms of therapeutic target. There are a number of drugs out there, but they're not as specific yet for this particular gene. But we do know that by silencing that gene, there is a functional difference, so a potential therapeutic target to be explored further. Next slide, please. One more. Perfect. So this is another gene as well that we were contacted by a team at University of Cambridge who were interested in this particular gene. And this is the one that is also has a number of mutations being detected with it. Next slide. And I think this just shows the real power of the cohorts that we have. So by us publishing and talking about the work we were doing, the team could contact us to include our cohort within this study. So together we could look at both adult and pediatric for DDX3X to look at it in terms of a functional way, but also as a potential therapeutic target. And again, on the side where we have what we call a lollipop plot, you can see there's mutations across the gene, and there's a number of functional studies that are still being carried out to look at whether DDX3X or DDX3Y could actually become a therapeutic target within the pediatric BNHL. Next slide, please. So basically what's next for our study, we've got a number of projects happening, and one is around the validation. So if to do any study, we have to have a validation. So we have shown p53 as a prognostic marker in a good sized cohort, of very well curated cohorts, thanks to the power of CCLG. However, we need to validate this before we can really take it forward. And to do this in the UK would take a very, very long time to do. It took almost 10 years to put the original cohort together with all the clinical information, and from the start of the study to the publication. And we didn't really have time to be doing that. We are obviously still collecting patient samples through CCLG, but our colleagues across Europe looked at their patient cohorts. So Germany have published their data sets, Italy and also Spain, and actually their research has really helped to validate kind of our original findings, and this has helped to expedite it to the kind of next level to kind of look adoption into kind of clinical trials. Next slide, please. And this is basically what we're doing now, is one of the big studies. So by pulling all these different cohorts together, we've got 774 cases, and this is really powerful. I mean, when you're talking about pediatric cancers like lymphoma, the numbers are very small, which does kind of impose a few challenges when looking for significance and really kind of getting that confidence that what we're finding is valid for clinical trials. So we've looked at 774 patients and basically evaluate that prognostic relevance. And we presented this at a conference in September, and you can see the kind of the wealth of people on that abstract and the different nationalities as well, which is fantastic to say, that collaboration. And really showing that p53 mutations that were first identified in the CCLG cohort are a really robust prognostic biomarker in pediatric BNHL. Next slide, please. But we also wanted to look at the care pathway analysis. So kind of patient-public involvement, engagement, and clinicians' interviews is a really key part of the kind of work that we do. Without understanding where the work fits into the care pathway and what impact it would have, but also what impact it would have on the patient, the parents, family, friends, it's a really important part of all the studies that we do. So as part of one of my projects with my PhD students, we did a number of interviews with patient and public groups talking about p53 as a molecular biomarker and how we'd like to translate that to the field. Next, please. We also mapped out the care pathway with clinicians at the Great North Children's Hospital. This is also a really valuable part of the study as well from a scientist's perspective. I'm not a clinician, I'm a scientist, so I don't understand all the care pathways, and understanding where your work fits into that and fits into the whole structure is a really valid part, because you can find out all sorts of things, but is it translatable is the biggest question, the so what question. We wanted to make sure that the work that we were doing and where it would fit in. Next, please. Also went through an ethical approval and design and conducted the interview. Next, the final part. And then we collated this and analysed this, utilising the expertise within the Newcastle in vitro diagnostic cooperative. Next, please. And just a summary of some of the findings. So when we asked about views on patients and family acceptability of the treatment, We got some really insightful comments back. I mean, he's just a few snapshots, but basically, I think they love to have the treatment that is successful, comes with significantly less toxicity burden and late effects burden. Next, please. Talking about needs for de-escalation of treatment. Again, it's sorry, I've copied the same thing in there, but basically, again, that is also supporting the de-escalation of treatment. Next slide, please. Talk about toxicity. The treatment package comes with a significant burden of toxicity and side effects. I think that all units across the UK are able to manage and deal with those, but a significant burden for the family. And that's come comment from one of the clinicians about how they manage it, but also that potential impact and how we could work together to resolve it. Next, please. And also talking about genomics, about how that impacts on the care pathway and the real enthusiasm because era of paediatric oncology where genomics is opening a doorway and basically on the threshold of quite a significant change. And you can see from other studies that CCLG support across particularly brain tumours and leukemias that you can see that genomics is coming out in terms of that risk stratification in trials and also the understanding of the disease more for identifying new therapeutics. Next, please. But also really pointing out the hurdles of introducing genomic biomarkers. And I think this in terms of kind of NHS and the kind of new 10-year plan and the shift to community, it's like, how do we make this happen? One of the hurdles is not getting the test done or getting it done in a timely fashion. And if it's going to take 4 to 6 weeks, particularly in a disease like Burkitt's lymphoma, you have a very small time frame between kind of if you're going to diagnose and relapse that actually want to get everything as early as possible. We don't want to wait 4 to 6 weeks. And how do we make that happen? And basically, it's not really about the diagnosis. It's basically getting the right treatment to the parents for the children and also that family to make sure that they're getting the right treatment. Next, please. So what are we doing next? So based on all the evidence that we have now and those studies, we're working across Europe internationally to across the European Intergroup on Childhood Non-Hodgkin Lymphomas, ICNHL. This is a cooperative group designed to really facilitate collaboration. So this is really helping to bring those studies together for us to analyse that data. Has members across Austria, Czech Republic, Japan, Netherlands, Poland, and there are 2 UK people who you may already be aware with who are very active within CCLG. So Amos Burke and Simon Bumpkin are involved in that clinical trial design. Amos is now was at Cambridge, is now with Birmingham, leading the clinical trials unit, and then Simon Bumpkin is MRC, clinician scientist who I work closely with at Newcastle. He's leading a lot of the BNHL within CCLG. I think there's some quite nice videos as well from Amos explaining about lymphoma and a bit more context as well. So this is what's happening next. The clinicians are taking this data and we're working together as scientists and clinicians to understand it to make sure that we design the right trial. Next, please. And this is my kind of penultimate slide, and it's really just to kind of take that moment to reflect. It's something that we don't get to do very often as scientists, but also to kind of really celebrate that what we can achieve when we work together to create that brighter future for young people with cancer. And with your support and all the scientists, nurses, clinicians, everybody who's been involved, fundraisers, and we've identified that first molecular biomarker in pediatric BNHL utilising the CCLG cohort, and that's going to be integrated into the next trial. We've also identified and investigated a number of potential therapeutic targets for people with BNHL in the UK and Africa that we're following up on. And the final slide. Yeah, I can't do any of this without all of these people. So we run the Northeast Lymphoma Research Group that is across Newcastle University and Teesside University. And that kind of covers the kind of spans the kind of Northeast region with myself, Simon Bumpkin, and also Chris Bacon, who is a pathologist and also a fantastic scientist as part of this team. Everybody who's contributed from the Lymphoma Genomics Group, past and present members, the bioinformatics, pathology, cytogenetics, UK collaborators, the UK cohort, Malawi cohort, all the funders, and finally a big thank you to all the patients, parents and fundraisers. And thank you very much for your time, and Ellie for being the slide mover honourer.
Yeah, I will apologise. I'm not sure I was the best slide mover honourer, but no, it was a really great talk. Thank you. It's quite amazing the scale like that research goes to across the world.
Yeah.
I don't think a lot of people know about. So it is now time for questions. We've not got any, which means you get one of the ones I scribbled down between moving slides to hopefully make people less shy. So you mentioned that the biomarkers, the TP53 markers, is going to be going into clinical trials. Will they just be looking at risk stratification, or will they also be testing new treatments?
There is research going on about new treatments. I think at the moment it's about that risk stratification. It's seeing that in the kind of current treatment about the looking at de-escalation and looking at kind of that higher risk group. Outside that trial, there is a lot of research happening, particularly with Simon Bumpkin, also Suzanne Turner. They're looking at different therapeutics around different Burkitt's lymphomas as well.
Okay, and my next question, if that's okay.
Yeah.
So in Malawi, it's malaria that triggers the cancer, is that right?
Sorry.
So
It's not well. It's a very complicated study, so there's a lot of controversy around that one. So yeah, malaria. I think it's whether or not it's an association or it is a trigger. The EBV is also part of it, but EBV is a lot of children in Malawi in that setting will have EBV infection. So there's different camps and controversy around it, but we feel like it's different parts of those infections that are making people more susceptible because obviously it's all linked to your immune system and your lymphatic system. But again, we're looking more at those biological factors rather than the epidemiological.
okay. Well, my follow-on question, which you can tell me is a bad question, but obviously it's forming the same cancer in the end, is do you expect infection to be involved in the UK in any way?
And we are also looking at kind of EBV infections in kind of UK, and there's some studies looking at kind of those EBV infections. And I think it does play a part, but I don't have the answers to that particular question. I think it's something which is still a big part of discussion and has been since Dennis Burkitt first defined it. So EBV does have a big part of it, and infection is around it. But in terms of the kind of most of the UK, it's more sporadic and looking at kind of mutation changes and those kind of abnormalities.